Everybody see my slide? Hello?
Yes.
Okay. So before we start, I would just like us to start with a short poll or quiz so that we can know areas to actually cover. So I'm trying to see how to create the poll. Okay, I think I have to continue. I'm not getting an answer. Okay, so I don't know if Mr. Fatoki can help me with that. So the big question or icebreaker is, what is bioinformatics? So this session would be dealing with introduction to biological databases and resources. But before we get into what biological databases are and what the resources available are, we'll first want to know why we need biological databases and also the resources available for the analysis. So the essence of biological databases is for research purpose, and the major harms that deals with computational biochemistry or biology is kind of like grouped into bioinformatics. So if you look at my slide by the upper right, you see a big elephant, and that's like scientists trying to question or to like give meaning to what bioinformatics actually is. So it's related to something like an elephant that has a very big part. You is so broad that you can't really give a particular word for what it means. So you can say, oh, bioinformatics could be like a palm tree, it could be the trunk, it could be like a pool, it could be different things as it relates to you. So bioinformatics is like all encompassing or joining several disciplines together, like immuno and chemoinformatics. So it's just simply the use of computer, that's bringing computer to life sciences. So through bioinformatics, we can predict tertiary structure of a protein or study genes and several other things. But it is just bringing together pharmaceutical application, chemical application, biological application, and then studying them with the use of computers. So that's just what bioinformatics is. I don't know if we've been able to get through with the pool of what bioinformatics is, so we could see like several other definitions of bioinformatics. Dr. Fatoke.
I'm still working on it.
Oh, okay. So, okay, we could continue though, but I just needed that so that we can proceed and then know several other parts to like tackle. So bioinformatics uses data derived from experimental model to answer research questions related to life science by the use of computers for biological analysis at molecular level. So it's just like, oh, the progress starts from you probably knowing, oh, there's a disease. So I could use diabetes for an instance. Oh, okay, there's this disease. How can I treat this disease? So if we want to look at the progression using just experimental, before you get to preclinical trial or clinical trial, it could get to over 13 years. And it's also capital intensive because at first you have to first screen. If you look at the standards, you have to first screen about 2,000 chemicals, or we could call them ligands. So we've not even gotten to probably like a protein you want to tackle or a particular gene you want to study. But we are looking at, okay, drug-based, probably like there's a ligand of targets that you want to use to tackle diabetes. So you have to first screen lots of ligands. So just as we're doing in lab, in phytochemical lab, I remember we could like do phytochemicals extractions. It could be ethanolic extraction or methanol, whatsoever as you have. So you have to go through those screening of about 2,000 phytochemicals. And then after screening them, you then bring out the its molecules. That's the one that you think will have highest bioactivity for the particular enzyme of targets. So once you've done this, you then also do some other screenings before it could get to like preclinical trial and all of that. So it's time consuming and it's also capital intensive. But with the aid of computers, it quite makes it easy right now, because with biological databases, we can screen over 1 million chemicals. So you have a large pool of ligands to screen. And then once you're able to come up with the its molecule, then you can begin to dock it with your particular enzyme of target. And then through that, you can study the structure-function relationship of the complex. So that's why we are talking about biological databases. So to start with, these are the available biological databases. So for protein or for enzyme, the best, or I won't say the best, probably like the common database is called PDB, Protein Data Bank. So this one represents, I don't know if you can see my cursor. Oh.
You can see it.
Okay, so this PDB means Protein Data Bank, but the J is like Japan. But there's a general PDB that you can, once you just type on your internet, you could just put in PDB or RCSB. So that's like a data bank where they load in crystallized protein. Okay, I will share the voting link now for the poll. Sorry for the break. So I shared the link for the poll right now, so everyone can just go to that link and then answer the question, what you understand by bioinformatics. I think everybody should be able to hear me. Okay, so it's called Protein Data Bank. That's where protein structures that has been crystallized are actually loaded. But another thing is, if your enzyme of interest has not been crystallized, you don't have to worry. There are ways to go about it, and we'll be dealing with that tomorrow. So that's like called protein tertiary structure prediction or homology models. So there are also databases that you can actually use for structural predictions. So we have like the Swiss model. I think I should have it in one of these pictures. So we have Swiss model or ITESA or AlphaFold. So those are available resources where you can predict a protein that has not been crystallized. So we'll do the hands-on tomorrow, and you'll be able to say how to predict protein tertiary structure that is not in the PDB. Then another one is the UniProt. So UniProt has almost, I won't say everything, but almost all the proteins that probably have not even been crystallized. So they have the structure, the faster structure of most, or will I say all proteins. So UniProt is like a best plug if the protein has not been crystallized and it's not in the PDB. So you can always get the faster sequence on UniProt. So you can always get the faster sequence on UniProt. Then another one is the PFAM. So the PFAM is protein families. So you have like more detailed information of proteins that has been crystallized and probably. Okay, so I'm seeing some definitions now. Some about 3 people said bioinformatics is the science of collecting and analyzing complex biological data, such as genetic codes. Then is interdisciplinary, computational tools, and all of that. So we can see that it has various definitions. So it could be genetic study, it could be structural biology, those that study protein structure and function also fall into bioinformatics. I mean, it also falls into bioinformatics, or can be called bioinformatician or what have you. So there are so as I was saying before, that's before I started seeing the responses. So more responses can still keep coming in as we go along. So this FASTA also is also another database where you have FASTA sequence of DNA and then protein. Then genomes is majorly for gene, BLAST is for sequence alignment. So this also has to do with probably if you don't have the protein structure crystallized yet, then you actually need to do homology model or protein tertiary structure prediction. So you have series of probably other organisms that have the same or similar protein. So you may have the faster sequences available. With BLAST, it can help you rank those faster sequences according to order of resemblance. To probably, if you want to study this enzyme in human, it will help you rank it according to order of resemblance. Then cluster W also does the same thing, but this does multiple sequence alignments. So you can BLAST several protein sequences, not just one. You can blast over 10, over 100, and then it will bring out the results in order of ranking of resemblance. So from there, you could be able to choose which one best fits your protein. Then NCBI also is like a best plug, and then PubMed. So PubMed is majorly for ligands. So if you're looking for particular small molecules, then you could use PubMed as, the site that has majorly every small molecules that have been worked on. There's also drug bank. If what you need is actually a drug for control, you can also use drug bank for that purpose. So biological databases could be divided into structure, sequence, and then any other thing. So structure-based will be the PDB, and then scope and CAT. So all of these are like for protein. PDB especially is for protein that has been crystallized through NMR or X-ray crystallography. So for the sequence, just as I said before, if the protein has not been crystallized, so you don't have to worry. What you do is homology model or structural prediction. So the database available for that can be the Swiss Prot, then the PIR, and the Tremble, ProCite, and then PFAM. So with all of this one, you have the protein sequences available. And then there's another one that I didn't mention, which is AlphaFold. So AlphaFold is also like a best plug for proteins that has not been crystallized, because what they do is also, AlphaFold is like a bioinformatics platform that they've used computational model to kind of like do the protein tertiary structural prediction of majorly all proteins. They are kind of like very vast. So if you don't get that structure in PDB, and then you don't want to go through the stress of using Swiss model or iTESA and the other databases, you can just first check on AlphaFold. If the structure is available on AlphaFold, then you are good to go, because it's highly recognized and it's publishable. So for nucleic acid, you have the GenBank, the DDBJ, the EMBL, and then others could be like Relibase, Rebase, and the other one. So majorly, biological database sources could be divided into three: the primary database, the secondary database, and then the composite database. The primary database is actually like a raw data that all you have is just probably the crystallized structure. Like for the PDB that I mentioned, that's Protein Data Bank. What you can get in Protein Data Bank is like crystallized structure proteins. So like they've not done any other experimental, or nothing has been done on it. So those are like primary databases. While the secondary databases, they've gone further to like do some other experimental things on this. Okay, like for proteins, they might have like done the NMR studies to give us the chemical chains and several things and how they are interacting, how the amino acids on protein are interacting with each other. And that's like secondary database. So you can other database to get that is like the EMBL. So you can get those type of information on EMBL, and that's for majorly for protein. Then you can also get that on EMBL, NMR structural informations. So that those will give you those specific informations of your protein. Then the composite database is actually more specific. So depending on what you actually want to do, and if several works have been done on that before, just like a bioinformatician might have worked on a particular protein, structural protein, and you're able to see the dynamics of the protein and how that affects its function, then it could be loaded into the database for other scientists to actually have access to it. So that's like the composite databases. Then, so this slide is just showing all I've said in picture in slide 3 in like written format and all that. So databases interfaces could be like the GenBank, EMBL, SwissProts, PDB, and the likes. Then for sequence alignments, you can use the BLAST and then the FASTA. So by tomorrow, once we get to the practical or the demonstration, we'll be able to like demonstrate how you can use BLAST to predict protein tertiary structure, and then also other homology model softwares that you can also use to predict protein tertiary structure if it's not yet crystallized. The multiple sequence alignments is the cluster, the multi-align or the di-align. While for gene study, you have gene scan, gene mark, and the likes. Then all these ones are more specific. They are like the secondary databases for protein. That's protein domain analysis and identification. So these are like secondary database for protein. That's PFAM, blocks, and PRODAM. So that will help you like to have specific information on a particular protein you are looking for. Then pattern identification and characterization. You have the Gibbs sampler, the Align AC, and then then protein folding prediction. So this is also like the composites, very highly specific. So if what you want to do is you want to predict how the protein will fold, and this is majorly for proteins that are not yet crystallized, either through NMR or X-ray crystallography or cryo-EM, then you can use predict protein or Swiss modeler or iTESA also to predict this. Then you can also use CLUST-Pro. CLUST-Pro, an advantage of CLUST-Pro is that it's going to predict the enantiomer, or if there are any other way that that protein could actually fold. Is going to give you over 100 predictions of protein folding. So CLUST-Pro is also very good software to utilize, especially when you want to predict protein folding. So important websites to note are these 3. They are not limited to these 3, but these ones are very important to. NCBI is majorly, you can use it for both protein or small molecules, EBI, and then SwissProts is majorly for protein structural alignment. Then ligand bases, I've been talking about majorly proteins or gene bank and the like. So for small molecules, the databases available are like PubChem, you have PubMed. PubMed has drugs and then other ligand small molecules that you can also like get. Then you can also use drug bank. And an advantage of this is that drug banks help you to like get your control. So for like I'll still use diabetes, you have metformin, which is like the general drug in treatment of diabetes. You have the structure there. You can also dock it into your protein, and then with the knowledge of structural biology that you now have, you can be able to like dictate or predict the protein or the complex interaction to see how metformin interacts with your targets in comparison to your new ligands or new small molecules that you want to screen for treatment of the same disease. So with this, I think I've come to the end of the lecture. So forgive them for questions if there's still time before the next speaker comes up.